It's titled Assembly of Hybrid Bacteriophage Beta DLPs is sensitive to extracellular osmolytes. Selection of marker matter.
Selection markers matter.
Selection markers.
So I apologize in advance for the difference between the title of my actual talk and the title that you all have. Between the time that I submitted that and presented this, I got more data which indicated my initial assumptions were completely wrong. And so I'm going to be presenting the good stuff to you now. As most of you probably are aware, in the FinLab, at least biologists among us, are interested in using viruses as nanomaterial building blocks for a variety of applications. In the past, we've focused a lot on the venerable cowpea mosaic virus, which has a long history of scripts. MD first started this work in collaboration with the Johnson Lab, and we really loved it because these particles were monodispersed building blocks, which were easy to work with. They've been extensively studied in the past, exceptionally easy to purify and very high yield. One of my favorite things in the lab was the smell of plant material and beta-mercaptoethanol in a blender. It's just the most relaxing thing I've ever done. And in addition, these were pretty cool because they have interesting potential as plant biotechnology agents. So the idea being that you could engineer this to be a vaccine, for instance, and then simply grow it in plants, and you just eat the plant and you have your vaccine. And I won't infringe on the Manchester Lab's work in vascular availability, but it's really cool stuff. The only problems that we had with this was that there was a long lag for the growth of the particle for production, and this problem of reversion of mutants where we would make a recombinant version of the particle, and while propagating in live plants, it would revert to the wild-type sequence. So that was really challenging, and it made it difficult for us to do chemical analysis of these particles. And so this is about the time that I joined the lab, and MG and I were talking, and we settled on working with bacteriophage Q-beta. This is great, phage all today, it's wonderful. Bacteriophage Q-beta is kind of cool. It was discovered, I think, if I recall correctly, in some sewage in Japan, and I have yet to translate the original discovery paper, but it's out there. The virion is exceptionally simple. It has one of the smallest genomes known, 4.2 kilobases, but the only thing that we're really interested in terms of this talk is the so-called A1 protein, the short version of which is 133 amino acids long, and when recombinantly expressed can form in the cells by itself these virus-like particles. You can see that there's some large pores. We can actually get small molecules in and out of those, which is kind of cool. But what's more interesting is that the bacteriophage takes advantage of the natural levels of TGA suppression by tryptophan to incorporate this A1 extension into the live virions. So previous work by Pumpkins and co-workers at the University of Latvia took advantage of the same principle. And what they did was they cut down the A1 extension to 6, 13, 19, 17, 72 amino acids. And relying on read-through, they were able to get incorporation of these extensions into particles. They also did some experiments just changing this to a glycine, but they didn't really carry it forward. All their subsequent follow-up papers used the unnaturals and scripts, use the TGA suppression methodology. But we were interested in something a little more specific or controllable. And so when I joined the lab, one of the first things I did was I cloned the short coprotein gene into standard pet vectors that just used T7 control for overexpression. Later on, when I was interested in making fusions, I cloned the coprotein with a fusion domain into a compatible PCDF vector that used an orthogonal selection marker and has convenient cloning sites for anyone who's interested in maybe displaying these on viruses. In the beginning, we cloned initially into a connomycin vector because we had just gotten a fermenter and we wanted to do large production runs. And because beta-lactamase is secreted into the media, it's not generally compatible with fermenter runs. You hydrolyze all your antibiotic and then there's no more selection for your plasmid. But in either case, it's the PMB1 origin of replication. When you induce co-expression with this system, you get these two genes produced and you produce virions that have your fusion domain on the outside. So here I've shown a 3-helix bundle, and I should probably introduce that a little bit better. And specifically what I'm going to be focusing on in this talk. This is the Z domain from Staph aureus protein A. Now I say from in quotes because it's actually a technologically modified version of the B domain, which binds to IgGs in solution. The Z domain IgG interaction is about has a 10 nanomolar Kb, and when displayed on N13 phage has a 50% saturation at about 20 nanomolar. So it's a pretty tight binder. Once I did the expression, I wanted to make sure that this was on the outside, not the interior of the capsid, and so a simple ELISA was in order. And basically, we laid down a random IgG that we had in the lab, the Z-domain particles, and the anti-Q-beta IgY, and finally detected the IgY with an anti-IgY enzyme. And you can see that the only thing that gives a response is the Z-domain particles, indicating that we have successfully displayed Z-domains on the outside and can bind antibodies, which is pretty cool in and of itself. But because we do a lot of engineering in the Thin Lab, we were interested in exploring the possibilities of these two different vectors and their two different selection markers in effects on the number of extensions per VLP that we got. And you can see here with the PET11, which uses the beta-lactamase marker, we got a little over 20, and the same thing with the PET28 vector, which uses kanamycin as a marker. So this was kind of cool. We could use these interchangeably and get the same results. However, our experience in the lab was that this was a little bit more variable with different fusions. And so, on the next one. So right here, I've just got an electropherogram of the different proteins, some different protein samples along the way of an expression. Our starter culture shows no induction, T0, no induction. After 4 hours of induction with IgG, we have huge bands for the co-protein, and in this case, it's a CMG2 fusion. That's capillary morphogenesis protein 2, important for anthrax. And so we were displaying that on the outside. And here, I'll black box the purification method, but you can see that we get a couple contaminating proteins. The major one, which is present in all these samples, is we analyzed by mass spectrometry, and we found it was this protein, outer membrane protein F, or OMP-F. A little bit of literature searching revealed that we could repress OMP-F expression by simply adding sucrose to the media. So with 5% sucrose, you suppress 60% of the OMP-F in these cells, and this works pretty fast. You don't have to add sucrose to your starter cultures, only your production cultures, and you decrease OMP-F production. So we verified this with our own experiments, and what I'm showing you here is a comparison between minimal expression media and minimal expression media with sucrose. And you can see these are samples of the T4 time point. The OMP-F to co-protein ratio drops significantly with sucrose, so we've saved ourselves a bunch of effort in the purification rounds, which we thought was fantastic. Unfortunately, what we noticed was that when you use the PET11 vector under these same conditions, you actually suppress incorporation of the Z domains into the particles. I'll pause a moment here to introduce these color codes. The blue bars are what are theoretically the greatest number of Z domains per particle that we could get based on the amount of expressed protein. And the red bars are what we actually observe after the purification steam. So this just gives you an idea of the lack of correlation between what's incorporated and what's produced. At this point, I was really, really interested in recovering my numbers, and so I thought, just for the heck of it, I'll try adding glycine betaine, which is one of the most potent osmoprotectants for E. coli that's known. And it turns out, fantastically, that adding glycine betaine completely recovers incorporation of Z domains into particles, which was all well and good for engineering's sake, but discovery biology indicates that we do more experiments to figure out what's going on. So I did a simple titration and figured out that the addition of 0.2 molar MECL doesn't inhibit growth of the cells at all. But when we do the purification, what we find is that sodium chloride actually increases the number of Z domains that we get per particle. And there's no growth inhibition here. The doubling times are pretty much exactly the same for these two samples. So we're not messing with the metabolic pathways at this point. But we do see a significant increase in the number of Z domains that we get on the particles, up from 20 to about 32. And in this case, you could argue that perhaps it's a result of increased translation of the Z domain, which you can see up here blows out my scale that I've been using. But for the most part, in this case, we see that increased translation ratios don't result in an increase in protein incorporation, which is we don't totally understand, but I've started to come to a conclusion about the sucrose system.